All dreams considered. All dreams considered. All dreams considered. Welcome back to All Dreams Considered. This is Sarah, and today I'm talking with Sid. Would you like to tell us a little bit about yourself?
Yeah, I'm sort of somewhere in the realm between amateur and professional mycologist. I'm based out of Eugene and do research at the U of O, independent, and with societies sort of everywhere. I basically got sucked into mycology after doing a microremediation project at LCC, Lane Community College in Eugene, and it kind of like just spiraled from there. They sucked me in very quick, and I find myself like every spring and fall traveling all over, just basically immersed in fungi.
Wow.
It's a blast. But I'd say research is definitely my main focus, just because like there's so many unanswered questions within mycology. Like it's a very young and rapidly growing field, and I feel like a lot of people don't realize how many avenues there are to go down in it.
Yeah, so I met you at the mushroom camp out at West Wind, and I was fascinated by so many things that you talked about. Not only your passion for hunting and finding mushrooms, and you're the one who told me to go look for the stinkhorn. I'll put a picture up so people can see what that looks like. Mm-hmm. That was amazing that I found that.
Yeah.
Well, actually, can you tell us about the stinkhorn?
Sure. It's kind of like a recurring guest at Camp Westwind. The scientific name for that one is Phallus hadriani, the purple egg stinkhorn. Really just a super cool group of mushrooms. You know, they always start off enclosed in these eggs. This one in particular is known for growing in dunes, which at Westwind, you know, there's a big old section of dune they have on their property, beautiful property. And so depending on the stage, you might find that purple staining egg that'll hatch open and sort of this really, I mean, phallic looking gnarly mushroom will pop out. And you know, they have the name stinkhorn for a reason. Once they're mature, like, we'll always bring it in at Camp Westwind, but
and
eventually
somebody takes it out.
Somebody moves it outside. It's usually like that first breakfast after it's been brought in, when everybody is like, what is that smell?
Yes.
And it's a really complex mixture of aromas too. Yeah, that's a perfect one. And it's that sort of brown slime called the gleba all over that honeycomb that's the source of the odor. And even just like the little bit left on that specimen, which it's meant to attract flies, essentially flies and beetles that'll eat it and then disperse the spores everywhere. And it's just a complex mixture of smells too. Like everybody who smells it tends to pick up something different. It's almost like a truffle in a gross way, I guess. Descriptions really range. And then it's interesting because there's always like one or two people out of a crowd that don't smell anything. Like it's, I don't know. Those complex mixtures are really interesting to me, especially when it's like odors, because it shows like sort of what the mushroom is offering and then also how diverse human noses are. It's always like, I don't know, they're a great conversation starter, and we've found it every year at Westwind so far, either the first or second camp, and I don't think I've actually ever been the one to find it either. It's usually like I'll mention it to somebody, or I'll be like, yeah, this is here, we need to find it, and then it'll end up on the table.
Yep.
It's a fun one though. Sometimes people will eat them in the egg stage before they hatch and the smell comes out, but I don't know about that. I've not really heard great things.
So, you mentioned going to school and, you know, finding this class, but were you interested in mushrooms before that?
I mean, I sort of grew up in so my mother was in virology and public health growing up. So I was always in a really like science-centered household, and I always had an interest in I spent most of my time outdoors given the chance. I just appreciated everything, but it wasn't until, oh gosh, I don't even know how many years ago at this point. It wasn't until right before I went to Lane Community College, while I was living in Cottage Grove, that I was just finding mushrooms everywhere. I walked everywhere around town, and I wanted to start identifying them, learning more about them, and I picked up the book Mycelium Running, by Paul Stamets, and it's really like a not only cultivation but really myco-remediation centered book. And it had a lot of simple techniques for like cloning and growing mushrooms. So it sort of timed out perfectly. After I started going to LCC, there was a spring undergraduate research symposium. And so I decided to do a myco-remediation project, essentially using oyster mushrooms, which are like a really aggressive white rot decomposer, to break down the paper towel waste on campus. And they do a great job of it, like very quick. The towels will get bleached and brittle as they break it down, and by the end you're almost just kind of left with this like duff that can immediately go in a garden or anywhere. And they allowed me, this is like where it really went downhill, or picked up steam, is they allowed me to use the life science laboratory to do the agar work and culture to create like inoculant and go through it all and run my tests. And I basically made friends with the two managers, Barbara Dumbleton and Rosie Kerwin, which shout out to both of them, they're amazing. They allowed me to basically continue using the facilities for mycological research. And so I sort of just like, while I was going through school there, was always running research out of that lab concurrently. And really it's like learning how many untapped avenues there were. Like there are it's a young enough field and there's so many different areas to go in that like even as someone really new to it, there's questions that you can answer. Even as an amateur new person into mycology, like there's just so many questions to ask that I found it wasn't hard to like run into areas where just nobody really knew something. And once I developed the skills to actually really start consistently cloning and growing mushrooms, like it's still my stance. I'd say I like mycelium more than mushrooms for the most part.
Interesting.
It's the part that does the moving, the eating, the mating, all the bioactivity. It's like really, it's the organism. And
because mushrooms are just the fruiting body.
yeah, just like one stage in the life cycle, a very brief stage. The mushroom might appear and rot within like a week and a half, release its spores, but that mycelium might have been there for hundreds of years in some cases. Old growth forests are like the honey mushrooms out in eastern Oregon. I think they clocked that one at over 1,000 years old. But really, once I learned how to manipulate and encourage mycelium, I just started cloning everything. I started doing like mating studies, microscopic studies, and like even something so simple as that, like cloning. We don't have photos or written documentation of what the mycelium looks like, even for common species. So like already that was kind of the first thing I got really into, was just like cloning new things, documenting the mycelia, and writing everything up, because like...
Are you writing a book?
I need to.
With all the research you already have, you could probably get somebody to even help you just put it together if you've already got all the...
It's definitely something I'm interested in. I mean, there's been so many different things where like I currently do research at the U of O, and I co-authored a paper at that lab on leafcutter ants, which are fungus farming ants. But like...
With the cordyceps, perhaps?
No, this is the symbiotic one that they grow on purpose. It's actually Leucoagaricus. So like, I guess one of the common names would be like the dapperlings or parasols. But that's a whole relationship. That the leafcutter ants is one whole subject. But like, I don't know, my PI there, Robert, will kind of joke that like all these projects I'm kind of doing in my free time, mostly just passion driven, not paid. Like I have a lot that's more or less like grad student level, PhD student level projects that I need to get out there. Certainly at least I have one paper that I'm hoping to get out in the near future, also on leafcutter ants from the U of O, but I need to start compiling and organizing things, because right now I just have like big old file cabinets and hard drives full of everything.
Make backups.
Yeah.
Good. Wow. So, can you tell us about other projects that you've worked on? Well, actually, first of all, how, I want to get to the other projects you've worked on, but I want to know, how does somebody go about learning how to do what you do? Is it I know you did it through school, but it sounds like you've got extra passion involved.
Absolutely. And I mean, I would say, really, unless you're in one of the few mycology-centered programs at a school, for the most part, you're not really going to get, there's some taxonomy you can get. There's, especially with sequencing becoming more common, there's curricula focused on things like that. But, a lot of it is stuff you have to self-teach. Or, you have to engage in the learning yourself to get these skills. For me, what's sort of, where I picked up my first set of skills regarding cloning and microscopy was actually through Facebook groups. I can't advocate enough, people have mixed feelings about social media at this point. A lot of people have evacuated Facebook, but for me, what drug me in is I don't really use it socially, I just use it for the groups. Because I was finding these massive international communities, mixes of amateurs, experienced growers, actual mycologists from the whole world over, and really a plethora of knowledge. You could see what questions other people have asked, you could ask your own questions, and then of course some of these groups have been going for years, so you can dig through the archives and see. And that's where I picked up sort of the foundational skills, and then LCC gave me the resources to execute those and get good at it. And really
what are the resources that you needed?
So for example, things like cloning and manipulating mycelium, it's actually pretty cheap and simple. Overall, probably the biggest tenet is actually shared just because kind of as a foundation for most microbiology when you're manipulating things on agar, like petri dishes of nutrient medium, is sterile technique and an azenic environment.
Okay.
So a clean space, and then the petri dishes, the agar, things like scalpel blades or loops, most of that's pretty affordable, especially now that there's companies catering to mushroom growers and mycologists. You can pick up a lot of these things for pretty easy, and then it's sort of a combo of trial and error and going through protocols. I think one thing that let me at least move pretty quick was that I was sort of, I've always been pretty autodidactic, I've always self-taught essentially, but I would use those Facebook groups, I would go through books, and also I pretty much am always just constantly reading papers, published articles on new methods, old methods, etc., to sort of just expand my skill set, learn more, and you can go way back to early papers on things like studying of mating and sexuality in fungi and see the methods at that time, and then you can see researchers bouncing ideas off each other, sort of this literature history of how methods have changed.
Wow. What's interesting to me about this is you mentioned that there are 30,000 different sexes that
probably more.
Yeah. Can you talk about that?
Absolutely.
I mean, we have. Actually, as humans, we used to have two, and now we're realizing that it's actually a spectrum for us. So it's interesting that mushrooms kind of led the way in that.
Certainly, they set an interesting precedent for how to look at things like genetics versus expression and diversity. And I don't know, I mean, really it's, especially when it comes to people's personalities and identities, you can see pretty quick that it goes a lot beyond just the genetics. Just the chromosomes or things like that. Especially with humans, it's really interesting because all the hormone dynamics there. But fungi,
do they have something similar like hormones? How do they identify out of the 30,000 or more? So
that's the really cool part. I guess I'll try not to get too...
I mean, Tinder for mushrooms must be amazing.
It's wild. And they basically have an advertising compatibility system that's pheromone based. So the main reason there's so many different mating types, which you can roughly equate to sex or gender, is I'll try not to get too nitty gritty with it, but there's basically in the tetrapolar fungi, there's 4 regions, 4 alleles, basically, if you think back to Punnett squares. And each of those, it's not just A or B or X or Y. There's many, many, many different combos, and the actual final mating type is a combo of all those 4 put together. So, you know, you could have like AABB, you could have like ACBD, like really quickly you can see how if you have hundreds of different possibilities for each of those 4 sites, it multiplies really fast. And one of the cool things too is like, one of the most well studied is a super common mushroom, the split gill, Schizophilum commune or Schizophyllum commune. It's everywhere. It's a cosmopolitan species. And they're actually still developing new alleles. Like their mating types are still coming and going. They have these genes set up in a way where they encourage mutation and recombination, so they're kind of always mixing and matching this. And basically what these two sites with 2 alleles each control is pheromones and then pheromone receptors. It's almost like a lock and key system. So one of those sites will control what it's advertising with, the pheromones it's putting out, and the other site will control what pheromones it's compatible with, what it can receive. So you sort of have like a 2-part system, because ultimately it's going to be 2 colonies coming together to mate and produce your final mushroom-ready colony, because they like sort of fuse. They're no longer individuals past that point. And like essentially they can approach, but they won't be able to fuse. They won't be able to form something called a clamp connection, which is vital to the maintaining the fusion, and they won't be able to produce a fruit body. Unless their pheromones and receptors match up, they'll be, it's termed incompatible, and they'll actually start fighting at that point and start competing.
What do mushroom fights look like, or mycelium fights look like?
It's brutal. It's, again, this is also why like mycelium is really my favorite part, because when you look at how when 2 colonies butt up against each other, you'll often get what's called a zone of inhibition, or like a line of incompatibility. If it's between the same species, like say there's 2 colonies of the same species fighting for food in the same area. They're already mated, they just want food and then to produce a mushroom. They'll meet each other and then produce what's called a barrage. So they'll basically pump out huge amounts of digestive enzymes, lots of things like melanin, so you'll get like a dark line that forms, it'll be visible, and just this gnarly chemical soup. And it kind of does 2 things. It'll kill the mycelium at both sides, so they usually reach a stalemate, and it also burns out all of the food and nutrients at that line. So it's almost sort of like how firefighters, like wild and firefighters will create like burn lines to keep flame from spreading. They'll
talk about burning your bridges.
Yeah, they'll burn out a little line of territory. And it's funny, we actually will, you've seen it, if you've ever seen like spalted wood, how you'll have like wood with like varying white, darker patches and then black lines everywhere. Every one of those black lines is where 2 colonies have touched and stained the wood while they fight. And it's not always black either. Some of them produce reddish or bluish lines. Some of them are like too good at it, and like when it's interspecies, some of them won't stop. They'll actually be like good enough to kill the neighboring colony
and eat them.
And eat them. Yeah, some of them like it's sort of in between a mold and a mushroom, but most would call it a mold because you don't really see the fruit bodies very often. But the species Trichoderma, it's like kind of just a green mold looking. You'll see it like under bark if you ever peel trees, and that's like a really, really aggressive one. It does eat wood, but what it mostly does is eat fungi in wood. And so it's strong enough to, in most cases, it might make that line of inhibition first, but then it'll kill enough that it can actually overrun and start eating its competition. And again, this is something like, even when the mushrooms aren't out, if you like lift a log and see the mycelium underneath, or like see a piece of split firewood, like these dynamics are going on all year round. You'll always be able to see what mycelium is doing the rest of the time when it's not producing mushrooms. Like if it's not producing a mushroom, it's out there mating, eating, fighting. Some of them will produce like crystal landmines in their substrate, like oxalate crystals that are basically sitting there waiting for another colony to come along and basically start dissolving the crystals, and it'll just kill everything in the area. There's all these different methods. Some like will coil almost like a snake around, like, the two hyphae, like the single long thin cells that make up mycelium. They'll meet, and then one will actually coil around the other like a snake to more effectively attack it.
Wow.
Yeah, like mycelium is really the most interesting part to me.
Yeah, it sounds like they need some ambassadors to like help limit this slash and burn tactic that they've got going on.
And that's, I think why, always, I'm really passionate about like talks and education and like workshops, because until you actually get hands-on with some of it or like get to look through a microscope, like a lot of this doesn't really, you won't really see it. You won't really be thinking about it. But at least for me, once I sort of crossed that bridge and got into it, like it's a whole other world. If you just sit down and like look at the bottom of the log, you can see so many interactions. And on agar, when you're actually growing them, I might be like growing them on agar to see them mate. I might be growing them on agar to see them fight. You can do things like you can train them to be better fighters. Like you can build up resistance to things like trichoderma by like breeding for their ability to fight.
I'm suddenly wondering about the ethics of doing that versus like dog fighting. I mean, I would love to see all dogs loved. It's just my personal opinion.
Well,
but I don't know that it's a good idea to help people take their aggression out that way.
Yeah, it's complex with fungi. It's I think a lot of people think of them sort of as like a little bit passive, when they're some of the biggest, most ecologically significant things outside. They're moving and shaking, and sometimes that involves full-blown wars between fungi. Like, they'll compete over resources, they'll kill entire trees, or they might support an entire forest because it benefits both of them. Like, it's easy to forget that they're volatile. There's a lot going on. They can make huge changes, even though it might be under the duff or in a log where we can't really see it. That's definitely why I like agar so much, because it's like this perfect little 2D environment where you get to see everything. It's all out there. It's always been a problem that mycelia grow in dark, opaque things where we can't really see what's going on in the wild. We've had to make a lot of guesses. It's also certainly led to us not realizing how big of an effect they were having in a lot of cases.
Well, as I mentioned, everything that I do is based on dreaming. And in one of my dreams, I already told you about the dream series where mushrooms saved the world, but there was another dream in which, and I just lost it. Well, mushrooms are just so prolific, and they have an intelligence that we really haven't tapped into. And I think that's what you're actually seeing, is that their intelligence is which chemicals to push out at which time, or pheromones, or whatever.
Or what partners you're picking. They'll absolutely select between, like, they might be compatible with two partners, but they'll choose the one that's the fittest or complements their own genotype best. They can, like, if one of them has a nutrient deficiency, they might choose a partner that's really good at getting that one nutrient. They'll solve each other's problems.
So, wow, that just opened up all sorts of relationship ideas in my mind about relationships I see with humans. Wow, that's fascinating how we can understand humanity a lot more just by looking at what's going on with mushrooms.
Yeah, I think we have a lot to learn with, like, kind of the field of biomimicry, like modeling things. Usually it's the context of like modeling technology after nature, but overall I feel like we could probably be modeling many more aspects of just humanity after dynamics in nature, because they've mostly had longer to figure it out.
And was it you that was telling me about the Japanese, or did I see that on YouTube, about how the Japanese were trying to figure out how to find the most optimum pathways between several new underground stations?
Yeah, there's been a number of labs that have worked on it, but that was using slime molds. So they're not actually fungi. They're basically giant amoebas. which is just wild to think about. But they're fast, like very fast. And the way they grow is really, really interesting. They're sort of like about the closest you can get to a decision-making like computer algorithm with like very, very simple means. That's what that study and others have only continued to execute, is like how something that, I mean, we would view as so simple is capable of like solving these problems, getting through these, and like adapting and learning. You know, other studies have shown that they can form essentially memory. They can recall things, make better decisions in the future based off of it. And we're still only learning how like some of this might even be passed down when they breed, like when they actually sporulate. There's certainly a lot of dynamics like that with true fungi too.
Wow. Wow, okay. So I already have some ideas for like places we could go with this next time if we do it next time. But you've, in your studies, let's, focus on the things that you've learned that you're bringing to these camps. Mm-hmm. You know, like when people want to learn more about mushrooms, you're there saying, look at all this cool stuff I've discovered. You want to tell us some of the cool stuff and not give away your trade secrets, by the way?
Sure. I mean, really, I've done workshops and classes on a lot of different things. I usually try to sort of choose things that I've been working on that I think other people will be interested in. And one big advantage to things getting more popular and just like people kind of knowing more at baseline is I've been able to share more of what I have to like people at every stage. Lately, and like at Westwind, I've been doing a lot of like pigment research on fungal dyes, which kind of goes hand in hand with my interest in fluorescent fungi as well. And so probably one of the things that's really been a hit with like people at all levels has been this glowing or fluorescent mushroom henna, or phaeolus henna, as I've been calling it. It's derived from actually a really common and popular dye species, Phaeolus schweinitzii, called the dyer's polypore, or butt rot by arborists.
That's not a very flattering name.
No.
Of course.
But it had been observed to be fluorescent. Like it'll be used for dyeing textiles primarily. Like historically, that's basically all it's been used for. There's been some things like watercolors or lake pigments, but
and I have some fabric that we dyed. Mm-hmm. One is pink and one is brown, because if you change the acidity, it changes the color. Amazing.
Yeah, no, mushroom dyes are so dynamic.
Yeah.
That's another thing where I got sucked in really quick.
Yeah.
The fluorescent property was something people had known about just sort of as a novelty, but I kind of like accidentally discovered it worked really well on skin and kind of ran with it. Before I knew it, I had like basically this paste that was perfect for classes, perfect for workshops, because you can just do all the designs you want on your skin, apply it like normal henna, and then rinse it off. You won't see anything in normal light, but if you view it under black light, you'll be covered in beautiful fluorescent designs. And that was like just another example of sort of where like random exploration and like interest kind of just produced something that nobody had really done before. Mm-hmm. And kind of like with the mycelial morphology and like cloning things too, like I've always really been interested in like finding new stuff, like things that people haven't really tried before or thought about in mycology, which is not very hard, I will say. That's always something I want to emphasize.
Can I show this?
Yeah.
So this is using the, what did you call it again?
The phaeolus henna.
Phaeolus henna. And then that's what it looks like. You can see there's nothing there, and then there it appears. Oops. Phone call.
Yeah, very bright under black light. It's a big hit too. I like took a bunch out to Oregon Country Fair, where that sort of thing is like immensely popular. I haven't really I don't know, my husband says I need to. I haven't really started selling it yet, because it's really just been a fun time making it, giving it away, and Like, other than a few random like extras I've had after classes and stuff, like it's mostly just been gifting so far, which is nice. But I don't know.
Maybe you could partner with somebody who would like make it for you, so you don't have to. You can just continue doing the fun stuff.
Yeah.
Because I think you have that kind of mind that's like just always, ooh, shiny.
Exactly,
and then it's like
yeah.
you figure out how to do it, and then you can hand it off to somebody that's like, ooh, I want to do that.
Yeah,
And then they just give you royalties.
I need to get better at that part for sure. I don't know. I've never been too much of a salesman or capitalist. I just like the research part.
I would argue that your stuff sells itself. I mean, anybody who was at the camp, everybody was impressed. At least everyone I talked to, everybody was like, oh my gosh, this is amazing. And I don't think you need to be a salesperson.
I mean,
here's a word from our sponsor at Inner Oasis.
Welcome to Inner Oasis, Mind, Body, Spirit. We're in the heart of historic downtown McMinnville, and we offer you inspiration, tools, and gifts for the soul.
I mean, it got me out to like pretty much the Phaeolus henna and then some other like fluorescent dye, where it got me out as a instructor and like workshop leader at the International Fungi and Fiber Symposium last year at Port Townsend in Washington. And like, I don't know, I was really happy how just kind of like I don't know, I was actually, and still am, like, I think I've only been doing pigment work for 2 or 3 years now, and I never would have thought that just experiments like that, which is something I mostly do for fun, like it's just passion-based, would get me out as an instructor to like this international event. And it sounds like it's probably going to get me out again to the next IFFS that's in the Canary Islands.
Wow!
Yeah.
Will they pay for you to go?
We'll see.
Oh, I hope so.
They covered pretty much everything last time.
Wow.
So fingers crossed, but I'll definitely be going either way. I can't pass that opportunity up.
Yeah, I have some friends on the Canary Islands. I'll tell to go.
I've never been.
I haven't either yet.
Yeah, it sounds like a cool spot. Not as many mushrooms at the time of year we're going, but apparently the lichen diversity there is unprecedented.
So that's interesting. When you were talking about going out at night and looking for the bioluminescence and the different fluorescence that we could see, lichens come up a lot.
A lot,
so
yeah.
do you study lichens as well?
Not in the traditional lichenology sense, I would say, just because, the same thing is actually true for mushrooms. Like, probably the biggest avenue of research in both is taxonomy. So identification, description of new species, reclassification, and like building phylogenetic trees. What I have been doing with lichens is pretty much all chemistry-based, mostly in the context of pigments. In particular, like one thing I've really been focused on lately is there's actually a really common lichen. I think the common name is like sunburst lichen, Xanthoria parietina. Super common in urban areas because it can actually grow off pollution, essentially.
Oh my gosh,
Yeah.
pollution like oil and...
Actually, car exhaust or the gas output from paper mills. In Eugene, at Alton Baker Park, there's kind of like exhaust runoff from the road, and you can see all the trees downwind from the road are bright yellow because they're covered in Xanthoria. It's basically able to take nitrogen oxides out of the air and turn them into protein, turn them into nutrients.
Wow, that's amazing.
It's lucky because those same gases end up killing a lot of other lichens. You'll definitely see like how polluted, like lichens are really, really good indicators of things like heavy metals or airborne contaminants, just because you'll see instant shifts in diversity. Like, the presence of that much Xanthoria tells you so much about the air quality there. And you can see too, like, if you look at it from above, downwind of like the paper mill near Eugene, where they get some of those same pollutants, there's just this yellow streak that matches the wind pattern off the facility because the Xanthoria is able to take advantage of it, but at the same time, none of our more sensitive native lichens are there at all. They're all put off by the pollution instead of benefiting from it. You won't really see them until you get into like deep, nice, filtered air old growths with just like pristine quality. Yeah, with that lichen, very common around Eugene. It contains an anthraquinone called perietin, named after the lichen, It's just so cool. As it is, in its native form, it's just a yellow, bright anthraquinone, just a nice yellow pigment. But if you do what's called an ammonia fermentation in sort of the dye world, you'll end up with this sort of like reddish cotton candy pink dye, and you can dye textiles that color, cotton candy pink. But if you put that yarn out in the sunlight while it's still damp, it'll turn bright blue. So it's a photoreactive dye.
Oh my gosh, that's amazing.
Yeah, and so you can actually, I've had success doing like photoresist like printing, like people can do with cyanotypes, where you know, you can like lay leaves or little patterns down on top of it while it's pink, put it out in the sunlight, everything but the cover parts will turn blue, and so you'll get like a cotton candy pink and cotton candy blue design.
That sounds amazing.
Yeah, and despite the fact that this is one of the more popular dye lichens, it's been used for a long time. Like, we have papers from like the 1800s that document use of this lichen for its reddish, pinkish dye. No paper has actually detailed what's going on chemically. So, just again, you know, so many unanswered questions. Despite the fact it's so popular, the identity of that first pink pigment and then what it turns into when it gets hit by light, we don't know either.
Wow.
So lately, I've been
This
really...
is just one of the many things.
Oh, one
lately
of
you've
many.
been... Sorry, continue.
Yeah, no, this has been kind of my current hyper-focus, just because, you know, it's been winter and liken's pretty much the only thing I can collect around Eugene. But it's also like, I've been accumulating new analytical chemistry skills. I've been getting more informed on like doing a lot of organic chemistry studies, and this seems like the perfect opportunity to like put those into practice, because you know, I feel like I don't fully ever learn something until I get hands-on with it. And so this has been a great opportunity. Like, I've been doing tons of like HPLC and TLC chromatography to like learn the identity of what's going on, and at this point I have a pretty likely possible molecule for it, but I might need like an actual grant before I can confirm it, or just to talk really nice to someone at the U of O to let me use their machine.
Wow. I'd love to help you do that. Well, I don't know if I'm the specific person that, you know, but maybe this will help someone. Maybe someone will see this and say, yeah, come on out.
It'd be great. Yeah.
You only need an hour, 12, right?
Something like that. I have most of the prep work done already, and I'm HPLC trained. I just need MS. But or if someone would let me use their infrared spectrophotometer too, that could probably do it. No, I have like basically, well, you know, it's science, so you never fully know until you're like actually there. But assuming most of my research isn't just totally down the wrong avenue, I'm pretty sure I've narrowed it down to just a few molecules, which is really, really exciting. I've been like emailing a few lichenologists that focus on lichen chemistry and just trying to get better ideas of like, am I on the right path with this? Like, does this sound reasonable? And it's funny because even people who specialize in this sort of thing, like they'll have their own like it's such a niche group, they'll have their own avenue in it that they could just talk endlessly on, but then one other little subset, you know, they'll be like, unless I have a grant for that,
yeah,
probably not going to touch it.
Well, this is such an amazing area that people can study, especially people that are wondering about maybe a career change, or people that are thinking, what do I do when I grow up? I mean, it's like a completely open field from what you're saying.
Well, and like, you know, I kind of like the end result, I guess, of like a lot of what I do is like, I want to try to work on stuff that is not only interesting to me, but that I do think other people might find is cool. So like, Xanthoria is nice in that way because it is a popular dye method. And in going through this, I was already able to create a method where normally you'd basically toss your lichen in ammonia and then wait like maybe a month, and it may or may not work out for you to get your dye. I was able to get a overnight method, often faster than overnight method, where you'll have dye same day and it'll be maybe 5 or 6 times as concentrated as a traditional method. And so it's like, I kind of will go down an avenue like this, hoping at least that'll have things I can like give back to the dye community. And the same thing with like the breeding. Like when I was doing all the mating and sexuality work at LCC, like a lot of that was, I was sort of on the side making sure that like some of the takeaways from that I could give to the growing community as advice on how to easily breed, easily do like pairings and lineages and like mating work with fungi with very little resources. Like I always want to find newer or simplified methods that other people can use too, because I also think it encourages people to like try to find out more on their own. Like I'm really, really big on the education there. Like I want people to know how accessible some of this stuff is, and it's, you know, most of the time I'm not getting grant funding for a lot of this, so I've had to learn certainly how to do a lot of it cheaply and easily, which I kind of like. But
well, and that could even be a a whole other thing that you can teach people on its own, you know, how to do this just from your home.
Absolutely.
In fact, that might be a fun podcast idea for you.
Well, it's certainly something like I've spent a lot of time on. You know, I primarily do all of my work on paper, which is like kind of outdated, but I like the tactile aspect, and I like to do a lot of sketches and stuff. But sort of like, I nearly brought the journal, but as soon as I get towards the end of one, I get nervous about taking it anywhere, because I'm like, if this gets wet or something. But really, like, it's just stuff I try to pack in. It's like, there'll be methods people think are too complicated to ever do at home, and it's like, just doing a little bit more to find out, like, oh no, you can totally substitute, like, chloroform that was in this method for, like, acetone, which you can buy as nail polish remover. Things like that. There's, like, I always want to try to generate substitutes or easier methods, simpler methods, quicker methods, and like it's really nice just sort of laying out these research paths and seeing where they branch off, like what comes out of it, and how many questions I can answer by the end of it. And always, like, you know, a lot of the mating stuff was a little too niche for most people in the field, but I still was able to get nice like breeding methods and educational like documents and infographics out of it that I was able to give away. And so it's like, it's nice to be able to kind of benefit everybody when you're working on it.
Yeah, this is something that I don't know that this is something I want you to focus on, but I could see you having a team where somebody manages a Patreon account to fund the work that you're doing.
I definitely need to figure that out. One thing I'm realizing is, because I currently have a Patreon, but I realized really quick, like I added a cheap tier, because I was like, well, I want this to be accessible, I want everybody to do it. And then I found myself running into these dynamics where I'm like, well, normally I would just post everything on Facebook, and then the more complicated or longer stuff would go to the Patreon. I found myself like not wanting to post stuff to Facebook, because it's like, well, what are the Patreon people getting if I'm just posting it all on Facebook for free? And I've sort of been like, you know, I got back from a vacation, I've been sort of rehashing a few things. It's been a tense few months, and I'm realizing like I sort of am a little paralyzed with the way it's set up. I think I'm probably just going to make it free, make it where if people want to contribute and donate, they can. But it kind of did the opposite. I thought maybe if I was making money off of it, it would like be more motivation. If anything, I just feel like a little paralyzed by it. Because I mean, I know the second I make it, like at least just add a free tier and pretty much open it up to everybody, like I'll feel comfortable posting everywhere, everything again. Unanticipated consequence. But you know, there's plenty of people, pretty much everybody who's stuck around to this point is like genuinely contributing. I mean, all the supplies for the Xanthoria project were through that. Pretty much like I've been able to actually afford things like TLC supplies, and it's definitely opened stuff up.
Yeah. What is your Patreon?
It's Moving Mycology Forward, Sidnee's Lab.
Okay. I'm making a note of that for later. Forgive my writing.
But no, it's really like, what I like to use it for is the more long form stuff, where like Facebook, I like Facebook over things like Instagram, because Instagram's really like, you get like a paragraph and then photos. Facebook is better for write-ups, but Patreon is where like I try to put long form, like really, really detailed all of it from the ground up, which was also kind of a problem because instead of posting super frequently, I would build up just these massive, giant things and drop them all at once.
Yeah.
I'm still learning how to do it best.
Do you have your own blog? Website?
No. I haven't known HTML in years, but
there's other ways.
yeah.
Yeah. Because I was just thinking that Facebook isn't something that you have control over, but your own blog, you just post everything there and then periodically put links up on Facebook, and anybody who wants to follow your blog, and then you don't have as much management, and you always retain your information.
Yeah, I mean that's definitely something I think would be a good move, and my husband is definitely more business oriented than me, and like I almost do this thing where if I get a little too overwhelmed by something, I'll just like go down a research path and like cut out, I'll stop doing paid work to just like focus on something I find interesting. Is sort of like just keeping my sanity. And you'd think that'd be great if people are interested enough in it to like give you money or funding, but it's almost, I don't know. I wouldn't say I'm avoidant of making money, but
I kind of actually, I like the way that you do it though. I think that is the wave of the future, that hey, I'm really interested in this. Who also is interested in it and who wants to support me going down these rabbit holes? And look, I can prove that I do this constantly. I mean, you've got all this evidence.
I'm considering almost just like scanning the notebooks and putting them all up because they're like information dense, so you get to see like basically all of it. There's probably some proprietary stuff that like past employers wouldn't want up there, but there's enough of it. Like I posted some of the Xanthoria pages just into one of the dye groups on Facebook, and I was surprised with the feedback. People were like, I was getting questions on like, well, how can I start my own dye journal?
Yes.
Like how, because I guess I never really thought about, because I've been journaling for a long time, like a really long time. I've always had journals, and I guess I didn't realize how that's a skill that a lot of people have to take time to develop. Like that sort of note-taking is like, I'm realizing something that people do certainly seem to benefit from seeing like how I lay my thought process out, because I almost do it sort of like in bricked out, almost like conversations with myself, observational reports, and then I'll have like blocks for summaries of an experiment, and then my summary of the experiment and like what I want to do next. And that note-taking style is just something I sort of just came upon. But the questions I was getting, like the feedback, like I think it helps people start their own dye journals, just knowing how to write it down. Because what is it? It's not science if you're not writing it down, I think is what they say.
Wow. Okay, we are at probably 47 minutes.
Already?
Yeah, I know.
Wow.
This is just flying by, and I want to be...
I'm sorry, I'll talk a lot and go on tangents given the opportunity.
So I actually love that, and that's why I'm trying to make some notes about other things I would like to interview you about. I would love to talk more about these, but before we do that, in the last few minutes, what is something that you really want to share with the world that I don't know, that's really inspiring to you right now? Because you've got the way that your mind works. It's inspiring to me. I'm like, ooh, how can I do this? How can I do more of that? How can I, and I'm not you, right? So I'm not going to do everything exactly the way you do, but being in the presence of someone like you is very inspiring to me. You know what I mean? It's so anyway, if you were to give advice to 10-year-old you, what would that be? I know that was a roundabout way of getting to that question, but I didn't know where I was going at the beginning.
I would almost say like probably what held me back for a while and like what sort of changed like at LCC was I guess realizing that things weren't inaccessible, like that you could actually do stuff. And like a big realization was like when, like in mycology, what I realized once I really started reaching out to people and seeing is like, if you see someone who's doing something you're interested in or has a lab you might want to visit or tour, like the passion's contagious. Like it all sort of seemed kind of this whole other world for a long time, but once I actually started like sharing my own excitement with other people, picking up on their excitement, and then just doing stuff and seeing what happened. Like, if I'd started doing that earlier, I mean, I can't even really imagine where I'd be at now, because it's already moved so quick. Like, you know, something like the dye stuff I got into, sucked me in, and then I'm teaching classes in other countries. And really, it's been that way with kind of like everything. I guess realizing, like, I don't know, that the world is sort of small in a way, but then also huge at the same time was really kind of key to it. I don't know if that makes too much sense.
It does.
But like, it all felt really inaccessible for a long time before I realized that, you know, science is definitely changing, but so much of it really is like the same, just exploration. Just make sure you write it down and talk to other people about it. It gives you opportunities to check yourself and others before you go just down a rabbit hole somewhere that doesn't make sense.
Yeah. I think if I was to sum up what you just said is basically we have the ability these days to empower ourselves to pursue pretty much anything.
I think, yeah, basically. And you know, the internet has harmed and helped that in certain ways, certainly, but like really, I don't know, I think that Gary Linkoff's quote is more or less just quit your job and devote your life to mushrooms. Just right now, quit your job and devote your life to mushrooms. And that's basically what happened to me, and it's only been through others' excitement, the community support, that like I've been able to get through that and still have a house. But like really, it is the sort of thing, and not just in mycology, but just everywhere. Like, I think if you really follow your passion and talk to a lot of people, and try to like get self-sustaining methods where you can DIY as much as possible on the cheap, things like that will get you surprisingly far. I never would have expected to be where I am with most of this. Just like if I look back at that first year at LCC, I would never have pictured all of this. I mean, at this point, I spent half the year traveling, doing classes, talks, like just immersed in what I'm excited about. It's great. Yeah. And you know, running into other people who get excited about this stuff is a huge part of it. Like you, things like this. I mean, it's shared passion that kind of gets us all together, and certainly that excitement, like the feedback, like talking to other people in the community and getting feedback on how excited they are. Like, I was able to use the method you made, or like, have you tried this yet? And then I get a new idea. Things like that, I think that's where a lot of the motivation comes from, is talking to people, realizing that others are interested too, and it's been great. Certainly, I'm just fully sucked in at this point.
Oh, I love it. I love it. I love that you're following your passion, even to the extent of, I don't care about money. I mean, I want to live well, but I think that's the wave of the future, is follow our passion. Mm-hmm. And thank you for all the work that you're doing and how you're just making incredible strides in this industry.
Well, thanks for letting me talk about it.
I would love to talk to you again.
I would love that,
okay.
Yeah.
Well, thank you, and once again, everybody, thank you for joining us. This is Sid. What's your full name?
Sidnee Ober-Singleton.
Sidnee Ober-Singleton.
Mm-hmm. Yeah.
Thank you so much for being here today.
Absolutely. Thanks for having me.
Yes, and once again, this has been Sarah with All Dreams Considered. All Dreams Considered. All Dreams Considered. All Dreams Considered.